Still is.
Okay, let's go ahead and start now. We're going to take a look. We're going to try and wrap up the chemistry chapter today. Maybe, we'll see. And the last thing we were talking about before this were the phospholipids, the cell membrane structures. It's what we use to assemble our double-walled membrane of our cells. We'll talk a lot more about that when we get into chapter 3. Now, the third category of the lipids, we've gone through the triglycerides, we've gone through the phospholipids. Third category are the steroids. The steroids are cholesterol. And we all know how evil cholesterol is. Cholesterol is, you know, it's like the monster of the food group. It's not really, it just gets a bad reputation. And the amount of cholesterol that we should eat in our diet varies every year by what study that is being supported now. So, but cholesterol is very important to our bodies. Cholesterol, these steroids, and that's what cholesterol looks like at the bottom there. It's a ring structure, and everything derived off of cholesterol looks like that kind of ring. Now, cholesterol and steroids make some very important chemicals in our bodies called hormones. These hormones, these steroid-based hormones, include our sex hormones, testosterone and estrogen. It includes aldosterone, a hormone that controls our fluid and mineral levels, as well as raising our blood pressure. Cortisol, which we use to deal with stress. It helps to regulate glucose levels in their bodies. We all thought it was just insulin that did that, but cortisol does that too. And then we have what are known as the androgens. These are the non-specific sex hormones, essentially. The androgens are also released from the adrenal gland, and they are, among other things, are the trigger for puberty. So these are some very important cholesterol-based structures. Furthermore, we use cholesterol as a reinforcing element in the structure of our cell membrane. We need, so cholesterol is not this evil chemical out there just lurking in the corners, waiting to kill us and cause heart disease and kill us right away. Too much cholesterol can be bad for us. Excessive amounts of anything can be bad for us. We use cholesterol, among other things, I forgot about this one, to make vitamin D. Vitamin D, we make our own vitamin D. When we get exposed to ultraviolet radiation, we make vitamin D in our skin. We need vitamin D so that we can absorb calcium in our small intestine. No vitamin D, no calcium. No calcium, we have weak bones. So milk, for example, is a good example of the best of both worlds. It gives you calcium from the dairy to help build up your bones, and vitamin D so that you can absorb the calcium and send it to your bones and keep them strong. So that's something else that we have to have our cholesterol for. And right here, our gallbladder. Our gallbladder stores bile. Bile is made in the liver. The liver makes bile to break down fats. When we have fat in our diet, the only way we can get it to break down, since it doesn't get along with water, is to chop it up in tiny little pieces. And that's what bile does. Bile acts sort of like dishwashing liquid on grease. It breaks it apart. And so it chops it up into little pieces that we can absorb. And so it's very important. Our liver makes it. Our liver, even if the gallbladder has been removed, our liver still makes bile. It's a common fallacy that if your gallbladder is taken out, you can never eat fat. That's not true. You can still eat fat. You just can't eat as much as you used to before, but you can still eat fat. Your body still needs fat. And the liver will still make bile to help break that fat down. Now, later on, when we get into AP2, and we talk about the digestive system, we talk about the reproductive structures, you'll see the similarity between cholesterol and all those other steroid-based hormones. They all have that same ring structure that you see on the screen, except for a few differences in the functional groups added there. There's very little difference in the structure on any of these steroid-based hormones. Now, the last category of lipids are called the eicosanoids. This is sort of a catch-all location. But eicosanoids do a whole lot of things. Probably the best one that helps us is something called a prostaglandin. Prostaglandin plays a very important role in blood clotting. When we clot, when you cut yourself, eventually you stop bleeding. As some doctors are prone to say, all bleeding stops eventually. Think about it. But we don't like it to go that far. We like to, we like a clot to form at the site of an injury. Well, prostaglandins keep that clot from getting too big and spreading out. If you're bleeding out from a cut in your finger, you don't want to have a clot that runs up your arm. You want to have a clot at the site of the injury. Prostaglandins keep the clotting actions from spreading. Inflammation is another one that prostaglandins play a big role in. Inflammation is if you've got a cut or a scratch on your skin and there's a little red area around there, that's a good thing. That says that you're healing the injury. When you have redness, we call that inflammation. It's caused by increased blood flow, which brings in more red cells that contain more oxygen. It's bringing in more glucose to feed the healthy cells. It's bringing in white cells to take out the damaged cells. So localized inflammation is a really good thing. We don't want to see, though, your hand be red up the arm from a little cut or a scratch. You know, that's not good. Prostaglandins prevent that from happening. Now, there are occasions when an infection may be out of control. And in AP2, I'll show you some very unpleasant looking imagery of hands and feet that have been infected, that are uncontrollably infected, where the inflammation is spreading across the foot and up the leg. These are individuals with diabetes that have, one of the characteristics of diabetes is a very poor circulation in the extremities and a lack of sensation. So a diabetic patient will injure themselves, not feel it, and not be aware that it's not healing properly. And their first indication that something's wrong is when they can smell it, because the tissue is rotting away, and because they have poor circulation, poor nerve sensations. They don't know, and it has to do with, and you'll see that this inflammation has spread, you know, across the whole limb. So we have a lot of wound clinics in Tennessee, locally, in fact, that specialize in these kind of injuries with your diabetic patient. So the prostaglandins are sort of a localized hormone, if you will. We often consider them a type of hormone. We consider eicosanoids a type of hormone. We'll talk all about hormones in the beginning of ANP2. So, which now leads us into the third category, the proteins. Proteins are all over the place and what they can do for us. Proteins can be structural. They can be globular. They can be structural in the sense of muscles. Muscles and tendons are made out of protein. So we can contract our muscles and do things. And that's good. That's how we move. That's how we pump blood. That's how we walk and talk and breathe. We attach muscles to bone using proteins. The tendons that are holding our bones holding our muscles to our bones is nothing but pure collagen. The most, as I've often said, the most abundant protein in the body. And it is the strongest protein. It is structural. It helps to hold us upright in position. It helps us to maintain our posture. It holds our joints together. Then our, what we call the globular proteins, are all of our enzymes and our antibodies and all the hormones that we manufacture. Those are all going to be not structural, but they're a functional type. So if you're a protein, you're made of carbon and hydrogen and oxygen with nitrogen thrown in there. Occasionally you may have sulfur or phosphorus, but usually you're going to always have nitrogen, because proteins are made up of what are known as amino acids. And a protein is nothing but a whole chain of amino acids linked together in a very specific order. So there are only 20 different amino acids. 20 different amino acids, but we can make thousands and thousands of different types of proteins. We can arrange the 20 amino acids in any order we want. We can extend that a chain of amino acids. We can keep using the same amino acids again and again. So our chain can be 300 or 400 amino acids long, maybe 1,000 amino acids long. The amino acids are held together by peptide bonds, and they simply, the chain continues to grow and grow and grow as it's needed to make a unique type of protein. Remember, there are only 26 letters in the English alphabet. And yet we have 300,000 or 400,000 words that we make out of those 26 letters. So 20 amino acids to make thousands and thousands of proteins is nothing. So now an amino acid always looks like this. It always has an ammonia group on the end. What we call the amino group consists of 2 hydrogens and a nitrogen. It always has what we call the carboxyl or the acid group at the other end. I mentioned this before, the carbon with the double bonded oxygen and the OH group at the top, that's an acid. So we've got an ammonia and we've got an acid. Coming off the middle carbon in here, I'm sorry, coming off of the other carbon, not the middle carbon, but coming off the middle of the amino acid molecule is what we call the functional group. This is what makes the amino acid different. That R group there, all 20 amino acids have a different functional group attached on the side. It determines what the acid is. So with 20 different amino acids, you're going to have 20 different functional groups that plug in there where we just say R. So, and they all end up doing different things. They all look like this. Now, the functional group that attaches there at the R can be a real long group or it can be a real small group, but your basic amino acid is going to look just like this. And we link them together with what are called peptide bonds to make our protein. Now, we often call a protein a polypeptide because it means we have lots and lots of peptide bonds. If we're looking at it as a polymer, a protein is a polymer because it's just a long, long chain of amino acids. These are peptide bonds. It's a type of chemical bond where if you're really, really interested, peptide bonds, when they form, kick out water. You can see up at the top there, the hydrogen and the hydroxyl group are being kicked out to form water. That's what happens when a peptide bond forms. And I dare anybody in healthcare to tell me why they were just talking about peptide bonds the other day. I don't think it happens that way. So, okay, now this is a little summary here on proteins.
Proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
I like how they just drop it there. But this is when a protein gets formed in the cell, the initial protein looks very much like beads on a string. Each one of those little purple balls there is an amino acid, and they're held together with peptide bonds, and it's just a long chain of amino acids. Now, proteins come in 4 structures. We call it primary, secondary, tertiary, and quaternary. And each type has a different shape. It assumes a different structure. So the primary structure is this, just a straight line, essentially. Proteins don't stay in the primary structure very long. They immediately, as soon as they get formed, they get shaped into whatever structure they're going to have. They may have, well, I'll show you in a second here, but we never see a protein stay in this position because it really isn't designed to function like that. So primary structure is a linear sequence of amino acids, straight line order. Doesn't, when it gets formed, it doesn't stay that way very long. Immediately it forms at least a secondary structure. The secondary structure twists the protein molecule around into, initially it could be what's known as an alpha helix, which looks like a spring, a spiral structure like our telephone cord. over there on the wall. You know, it's going to be a point in time where, you know, we don't have telephone cords, you know, anymore. So, but as long as I've got it, I'll use that as the example. But it's a spiral structure. It's a string shape. Where would we see an alpha helix? Well, the elastic protein, remember from lab, elastic protein, it can stretch and then come back to its original shape. Well, that's in a helix. Our hair is all protein, and it is also in this alpha helix structure because our hair can go through all sorts of shapes when we, you know, however we, you know, whether we wake up with bedhead or not, you know, or we brush it and straighten it out, it will always go back to its original shape because of the protein structure there. That's one of the types. The other secondary structure is called the beta pleated sheet. A pleated sheet structure looks like this. It's folded with that type of shape. You know, so it's you know, pleated means there's folds in there. The pleated sheet gives us great strength because we have all these proteins lined up side by side by side. Collagen fibers that make up the tendons have tremendous strength, and they're organized in these straight lines. They have a pleated sheet structure in place. So the collagen in our skin, the collagen around our bones, the collagen that connects our tendons are all pleated sheets. We see a good example of this about this. This is the time of the year you start seeing really large cobwebs forming, spider webs. And you see these really big black and yellow spiders. Anybody seen one lately? One of those orb spiders? And it looks like they're writing something on their web. You know, that their web, those spiders, their webs are amazing because they can withstand all sorts of stuff in the fall. That's when they show up, and they can absorb rain and storm and wind and stuff like that. And they're still there because their spider webs are made out of pleated sheet protein. So, and it just, no matter what happens, they seem to like be attached to a fence post or wherever, you'll still see them. You know, it takes a lot to get rid of that because the webbing is so strong. So, tertiary structure is simply a protein that has multiple versions of the alpha helix and the pleated sheet. You can see this often in a large protein. And you know, hemoglobin, for example, has lots of these structures embedded there. But hemoglobin itself is even more complex. Hemoglobin, the protein that carries our oxygen in our blood, is a quaternary or four-sided structure. It's the most complex structure you can have. It's made up of 4 clusters of tertiary structures with secondary structures built into it. A hemoglobin molecule is about 360 amino acids in length in a very specific order. So, in fact, here's what it looks like. There's the primary structure at the top, straight line, but immediately after the protein is formed, it is changed into whatever shape it needs to have to function. Many of these proteins have to you know, if they have to land on a target cell to do something, then they have to have a shape for the receptor site, like sort of like a key going into a lock. You can use your the key that unlocks my office doesn't unlock that door, and the key that unlocks that door doesn't unlock the door across the hall. You know, so it takes me 3 keys to get anywhere on this end of the building. So. And none of them unlock the outside door. I need another key for that because the lock mechanisms are all different. So you have to have a specific key. For proteins to do their job in a lot of places, they have to have a specific shape. They just, you know, they have to be bent around. And one of the advantages of the pleated sheet or alpha helix or the tertiary structure is it bends the protein so it fits where it has to go. It fits the lock on a cell, if you will, or it shapes the protein into these long strands of collagen fibers, for example, for us. Okay. The fibrous proteins are probably the most common. These are the very stable proteins. These are muscles. Skeletal muscles are all made of contractile protein, which are fibrous in nature. They don't change their shape if something happens to them. If they become too hot or too cold, if they get exposed to changes in pH, they're going to keep their shape. Keratin, the waterproofing in our skin, elastin, the elasticity in our skin, the collagen, I couldn't resist saying it again, the single most abundant protein in the body. I promise you I'll put that on a test question too, so that I say it enough, so. And certain contractile fibers, well, that's all of our skeletal muscles and our smooth muscles and our cardiac muscles are all fibrous protein. The thing about fibrous protein is that they're very stable. Even if they get too hot, they're going to have the same shape. If you cook a piece of chicken, you'll still see the muscle fibers in the chicken, whether it's raw, whether it's fresh, it's raw, or it's cooked. The same thing on a piece of beef or fish or pork or anything like that. It's all going to have the structures haven't changed. You could take them and look at them microscopically, and they would look just like they did when if you took a live sample out of an animal or a human. If you did a biopsy on a human and took out some muscle cells to look at, they'd look the same, whether the person is alive or whether they're dead, and you're looking at a preserved specimen. So they don't change. These are very, very stable. These are the fibrous proteins. Now, the other category, though, are the globular proteins. They are, they're round. They look like a little bubble. They're soluble in water. They are very sensitive to temperature and pH. They have very specific regions on their structure that do things. These are where you really run into the lock and key approach. Your antibodies that are formed when you're exposed to foreign antigens. Now, an antigen is a marker. It's a protein itself. An antigen is a marker on a cell telling you what it is. Now, all of our cells have antigens on them saying, hi, I'm me. Don't do anything to me. And our immune system doesn't attack that particular cell. On the other hand, a foreign cell, a virus, which is not a cell, a virus is not alive, but a virus has a protein coat and it has antigens sticking out of it, or a bacteria, bless you, or a bacteria, that just sounded painful. A bacteria has antigens on there saying, I'm not part of your body. I'm foreign. And whether it's harmless or not harmless, your body will say, you don't belong here, I'm going to kill you. It's pretty much cut and dry. I mean, after all, we have cells called natural killer cells, so they're going to go out and kill things. So the markers on foreign cells or foreign substances are antigens, and our response is to make these proteins called antibodies, which will glom onto the antigen, and they'll block its action. They'll block its action, they'll prevent it from functioning, or they'll mark it for destruction by the natural killer cells. So we make lots of antibodies. If you had COVID, you generated antibodies on your own. It didn't matter if you had a vaccine or not, you produced antibodies. Antibodies give you a natural immunity to that particular disease. Every time you have a cold or a sore throat or a viral infection, you end up producing antibodies so that you don't get that same infection again. And what happens is viruses cheat. Viruses keep changing their structure around a little bit so that their antigens are just a little bit different every time. So you may get the same, what you think is the same thing that you always get, the sore throat and fever and headache and nausea and vomiting and diarrhea. But it's not, it's slightly different. So, but our antibodies will respond to all the other ones. If you had, say, if you failed to get a vaccination for chickenpox, and you developed a chickenpox, it's a virus, and you got over it, and you were uncomfortable for about 10 days, and then you got over it. You have a natural immunity to chickenpox. Now, you may get it again because your immunity levels might not have been, you might not have had as many antibodies as you should have. So it's entirely possible that in some cases you may get it a second time or even a third time, but those are really, really rare. Those are way out there at the end of the curve. They're the anomalies. You know, most people that didn't get a vaccination for chickenpox and then develop chickenpox are usually immune the rest of their lives because you made these antibodies. That's what protein, you know, these globular proteins, things like antibodies do that for us. Hormones are a globular protein. All the different hormones. Hormones are chemical messengers telling us to do things or not do things. The reason we have to pee or not have to pee depends on a hormone called antidiuretic hormone. Antidiuretic means it prevents us from peeing. You know, diuresis means to be constantly urinating. Antidiuretic or antidiuresis controls how often we pee. And so our bodies regulate when we are going to release urine from our bodies and when we're not. It's a protein. It tells us, it regulates our fluid levels. Hemoglobin is a protein. It carries oxygen. One hemoglobin molecule can carry 4 oxygens for us. These we have hormones for growth hormone. We have hormones that stimulate production of sperm cells and egg cells. We have hormones that regulate that produce. Let me rephrase that. We have thyroid hormone, which regulates our metabolic rate in all of our cells. And these are all proteins. And we have enzymes. Enzymes control chemical reactions. Enzymes make chemical reactions go faster. Remember, we talked about that briefly. An enzyme is a protein that simply brings 2 reactants together. 2 reactants. You know, normally, you know, in a normal reaction in a beaker, you would throw in 2 reactants, and depending on the concentration of the reactants, they may or may not interact quickly. Or, you know, they may interact quickly, they may interact slowly. And they depend on just sort of randomly bouncing into each other to have the reaction. What enzymes do is they grab the reactants and pull them together. And so in our bodies, we can't do much to make a chemical reaction go faster. Chemical reactions will work faster if the temperature goes up. Chemical reactions will work faster if we have, if the particle size, if we have very small particles because they bounce around faster. And if we increase the concentration, well, in our human bodies, we can't increase the concentration. We're stuck with the amount of reactants that we have. We're stuck with the same temperature and the particle size is already very tight. So there's not much we can do to make a reaction go faster except produce lots of enzymes. Enzymes go to work causing reactions to occur by simply grabbing the reactants. We can flood an area with the enzyme. It grabs reactants, very tiny reactants in small concentrations that are moving around at body temperature. We can grab them, bring them together, and cause a reaction to work. This animation here shows you what happens with starch. In our mouth, we have an enzyme known as amylase. Amylase breaks starch down into sucrose, or actually into glucose. it breaks it down to sucrose, and then breaks the sucrose down into glucose. What it does is the amylase grabs the onto the starch and rips it apart. And so what was a large molecule before now starts becoming a smaller and smaller molecule. And when we get to sucrose, 2 carbons, the disaccharides, we break that down to glucose. The process of digesting carbohydrates actually begins in the saliva in our mouth. Amylase works very well at 98.6, 37 degrees Celsius with a pH of about 7.3 or 4 in our mouth. It won't work in our stomach where the pH is about 2 and the temperature is about 99 or even higher. Amylase is very sensitive to temperature and to acidity levels, but so it works very well right up in here. We start breaking down the carbohydrates. It's often said that if you took a cracker and stuck it in your mouth and just let it dissolve, that you would taste the sugars as they were being released from the starch in the cracker. Every time I've tried that, the cracker usually is dissolved away before I can taste anything sweet. But in theory, you should be able to taste the sugar as the starch is being broken down by the amylase. That's what enzymes do. Enzymes bring reactants together to break them apart or put them, they bring reactants together for a chemical reaction to take place. Now, these globular proteins are very sensitive. They don't like changes in temperature. They don't like changes in pH. In fact, if they're exposed to a high temperature or a low temperature or to a pH that's out of their range, too acidic or too basic, they become what we call denatured. They unfold their shape. They can no longer function. They no longer have an active site on there for reactants to land on because they've unwound. The active sites are deactivated. Example, here's an egg. Egg has a yolk in the middle, which is filled with cholesterol. And the white of the egg is albumin, which is clear. When you open up a raw egg, you got the clear albumin with the egg yolk, and you cook it, and the albumin turns white. It has become denatured. It changes its structure and becomes white. Now, there is a level where if we had a living cell, then if we had a temperature extreme or a pH change, then if we went back to normal, then the cell's enzymes would recover. Even the globular enzymes would recover their original shape as long as we go back to homeostasis, if you will. I haven't said that word for about a week, so that's not to say it. So anyway, if the change is extreme, however, we can't go back. You can't take an egg that has been fried up. You can't take the egg white, which is still albumin, but you can't reverse it. That change is, the damage is irreparable. You can't do anything about it. In our bodies, as we go through temperature changes all day long and pH changes all day long, anything that happens to the enzymes, they tolerate it and then they get over it, or in some cases they get significantly damaged. Consider what happens in a person that has cardiac arrest. Cardiac arrest, the heart stops. It's not pumping blood anymore, not sending oxygen out to the cells. However, the cells are still alive. Brain cells will last for 5 minutes. Muscle cells will last for up to 12 hours. Even after the heart stops, even after the brain is dead, the muscle cells are still going to be processing glucose, generating ATP, and releasing carbon dioxide. Now, what happens when all these muscle cells in our bodies are releasing carbon dioxide? It diffuses into the blood. and it makes our blood more acidic. Now, normally, when our body is alive, our skeletal muscles are busy producing the bulk of our carbon dioxide. The carbon dioxide goes into our blood. We pump it up to the lungs. The lungs allow CO2 to diffuse out, and we breathe out carbon dioxide, and we're happy. Well, we're not pumping blood, so the CO2 accumulates in our bodies, in our blood, and it makes our blood more and more acidic because carbon dioxide combined with plasma forms carbonic acid. And our pH starts dropping. So on top of the fact that the patient's heart has stopped, now they're becoming more acidic. Yeah. Now there's a whole lot of oxygen in the blood. I mean, even brain cells can survive for 5 minutes. Every pass through the body every time a red cell makes the circuit through the body, and it takes about 20 seconds to go from the heart back to the heart, to go through the whole body. Every red blood cell only gives up probably about 15% of its available oxygen. There's a lot of oxygen in reserve. It takes a long time, relatively speaking, to get rid of all the oxygen. It's not like the heart stops and you're instantly, you got 5 minutes or so to play with. The idea, make sure the thing is to keep the heart pumping. If you have to do that artificially, you keep the heart pumping. However, if you're not doing at the same time, some sort of artificial compression is never as efficient as the heart is. And so you're always going to be developing more and more CO2. That was a long way of saying one of the protocols you go through with cardiac arrest is to administer a large volume of baking soda solution, sodium bicarbonate, to your patient to neutralize their acidity. Large volumes of bicarbonate, plain ordinary old baking soda solution. So it's almost like a couple scoops of baking soda into a glass of water and stir it up. Well, no, you administer through an IV or direct injection into the blood, under pressure perhaps, because you want to neutralize all that carbonic acid that's forming. So anyway, we can prevent, catastrophic results up to a point. Anyway, okay. How does an enzyme work? Well, the enzyme has what's known as an active site. And the active site in the enzyme is where a chemical reaction will take place. We call the reactants substrates. I don't know why we just don't call them reactants, but chemists call them substrates, so we call them substrates. The reactants land on the active site. Now, the active site on an enzyme has a very specific shape. You'll notice the shape of the substrates, the reactants, match the active site. Nothing else can land there except those reactants. There is no other reactant. There are no other reactants that will fit there. Everything in our bodies will bounce up against every cell. Anything in solution will come in contact with every cell in our body, but unless there's a location there that fits that chemical, that reactant, nothing's going to happen. For example, consider this: thyroid stimulating hormone. It's a protein. It stimulates thyroid stimulating hormone. You want to guess what organ it acts on? Thyroid stimulating hormone. Just take a guess. Thank you. Yes, the thyroid stimulating hormone. I know it's Monday. It only works on thyroid stimulating hormone. It's a hormone released from the pituitary gland in the brain, and it only acts on the thyroid. But it will come in contact with every other structure in the body. But the liver doesn't have a receptor site for thyroid stimulating hormone. The kidneys don't. The bladder doesn't. The ovaries or the testes don't have anything for thyroid stimulating hormone. Only the thyroid gland in our neck. Now, conversely, after the thyroid is stimulated to make thyroid hormone, that will come in contact with every cell in our bodies and stimulate every one of those cells to increase its metabolic rate. So the active sites are determined by their shape. An enzyme that controls, the production, the breakdown of starch to glucose has active sites that only a carbohydrate molecule can land on. An enzyme that stimulates production of thyroid hormone will only have active sites on the thyroid gland. This is how we say that enzymes are biological catalysts. They work by allowing reactions to go faster. They simply bring small molecules together. All they're doing is putting them literally in close proximity. They're right next to each other. And when they're right next to each other, a reaction can occur. There's nothing special about that. We don't raise the temperature. We don't raise the concentration. We don't have smaller particles. We just bring them together. And when they're close together, like we see here, then the reaction occurs. We can make a new product that's bigger, or we can take a big product, big reactant, and turn it into smaller products. And so the enzymes don't get changed. The enzymes don't get destroyed. The enzymes get to do this over and over and over again. This is how they make chemical reactions in our bodies work. They don't get destroyed. They do it again and again and again. So now if you're an enzyme, you have a protein portion and a cofactor. The cofactor is often a vitamin. We often call it a coenzyme. It could be a metal ion like copper or something like that, or nickel or magnesium, but frequently it's a coenzyme, which is a vitamin. Vitamins help certain reactions to take place. The fact that vitamin D allows us to absorb calcium in the small intestine. Vitamin D combines with enzymes that will allow the calcium to be absorbed. In the cells lining the small intestine, there are probably active sites there that require vitamin D to be the coenzyme to allow the calcium to combine on the active site. That's what it does. We'll talk about this in chapter 24. Coenzymes are not wannabe enzymes. They're just a portion of the overall enzyme. That's where vitamins come in. Enzymes are extremely specific, though. One enzyme can't do anything except that one type of active site reaction. The substrates are very, very specific. Amylase only does starch. There are oxidase reactions. There are hydrolase reactions. Every time you have the ending ASE, you're dealing with an enzyme. So hydroxylase, oxidase, amylase. There's all sorts of enzymes. They all have the ASE ending. That's how you know it's an enzyme. And the classic answer is, what do enzymes do? They lower the activation energy so that a chemical reaction can take place. Instead of having to bounce around rapidly and all over the place, two reactants can come together quickly at a lower temperature, body temperature, at a lower concentration. So.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
He was so perky for just 2 sentences. But that's what they do. All of our chemical reactions that take place in our bodies are controlled by enzymes because we can't raise our temperature. We can't increase the concentration, and we're already dealing with very tiny particles to begin with. So enzymes are our body's workaround to get chemical reactions to occur. And if you are visual, this is, if you want to look at this again and again, this is what an enzyme does. This is how we produce amino acids into proteins. We simply use enzymes to bring, for amino acids to link together, form a peptide bond, and then get released as two proteins together. Two amino acids together, a dipeptide, which then may react with another enzyme to produce a second dipeptide, and now we have 4 amino acids linked together. So, and then the enzyme lets go and does it again. When the enzyme gets denatured, what happens is that the active site on the enzyme is altered either by temperature or pH. If we heat up the enzyme, the active site changes, the reactants can no longer fit there. And so if the denaturation is permanent, you end up with the damaged active site. If enzymes are damaged beyond repair, then this is what's going to happen, and we start having problems. Now, so that leads us into the last category of organic molecules, the nucleic acids. The nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They are the largest molecules in the body. They are the DNAs and the RNAs. Remember, a DNA molecule is about 6 feet tall, 6 feet long. So it's each individual strand of DNA is about 6 feet. So I'd be right about here, I guess. I don't know. Every nucleic acid is made up of what we call a nitrogen base, a 5-carbon sugar, which is a pentose sugar, and a phosphate group. And we're either DNA, the deoxyribonucleic acid, or the ribonucleic acid, the RNA. Now, the nucleotides all have the same 3 basic structures. They have a phosphate, they have a sugar, and they have a base. And if you look over here on the right-hand side, here is your phosphate group right here. Your phosphate is attached to one corner of the 5-carbon sugar. It's a pentose sugar. It's shaped like a pentagon. And so you have your phosphate attached at one end and your nitrogenous base on the other end. So you've got a phosphate, the sugar, and the base in this arrangement. We use the sugar and the phosphate for structure. The bases are arguably the most important part of the nucleotide, because when the bases are arranged together in a DNA molecule, the order of the bases becomes very important. The order of the bases determines the order of amino acids in a protein. DNA is a cookbook for us. It tells us how to put together us. The instructions in the DNA on how to assemble every protein in our body are based on the order of all these bases for every molecule, for every protein. The order of the bases determines the order of the amino acids in every protein we've got. And so these are actually instructions on how to put us together. So this is why we call it the genetic blueprint. The DNA molecule is a double helix mean, it's like a spiral staircase, like it's like a ladder that's been twisted. The nucleotides, in this case, there are the four that has the nucleotides use 4 different bases in DNA: adenine, guanine, cytosine, and thymine. These are the 4 bases we have. Now, the bases pair up with each other into what we call complementary pairing. Adenine always pairs to thymine. Cytosine always pairs to guanine. Well, maybe usually always, because there are mistakes, but we'll get into that when we talk about protein synthesis in chapter 3. However, right now, just say adenine always pairs to thymine and cytosine always pairs to guanine. In the DNA strand, which is a ladder-like arrangement, we have, if we have cytosine here, we're always going to have guanine on the other side of the ladder. If we have adenine here, we're always going to have thymine. If we have thymine, we're always going to pair to adenine. Now, the bases are held together with hydrogen bonds. The order of the bases determines the order of the amino acids in a protein. You have a long chain, a long protein chain. It's made up of individual amino acids. The order of the amino acids is dictated by the order of the bases. So there we see a DNA molecule, drawn much better than mine, but you can see the pairing, adenine to thymine, guanine to cytosine. And what happens is that the molecule develops a spiral structure because the hydrogen bonds that are occurring between the adenines and thymines and cytosines and guanines cause the molecule to twist. It's simply the effect of the hydrogen bonds. Now, what's important here, the 4 bases, you got to remember the 4 bases: adenine, thymine, guanine, and cytosine. Now, I likely in any kind of healthcare that you encounter, it's probably unusual that you have that kind of discussion. But this is one of those little factoids you need to carry around in your head. Adenine, thymine, guanine, and cytosine. These are the 4 bases here. And the bases are important. The order of the bases determines the order of the amino acids that go into the production of a protein. And the order of those amino acids is critical. Consider hemoglobin. Hemoglobin is 360 amino acids long. There is a genetic condition that is a homozygous recessive condition. It is 2 recessive genes. If you inherit both recessive genes from your parents, you would have this condition. This condition is sickle cell disease. To have sickle cell disease, you have to have a recessive trait from your mother and your father. You don't catch sickle cell disease. And in sickle cell disease, the hemoglobin is defective. The hemoglobin is brittle. The hemoglobin, instead of forming a nice 4-sided structure, when a person increases their demand for oxygen, the hemoglobin will twist into this crescent or sickle shape and cause the red blood cell to become crescent shaped or sickle cell shaped. And it becomes very brittle. Red blood cells have to be able to pass through very tiny vessels in our bodies. It has to be very squishy. And when it gets brittle, it doesn't, and it hurts. And it also reduces its oxygen carrying capacity by about 80%. Now, what's this got to do with amino acids? Well, it seems that in hemoglobin, in the number 6 spot, now remember there's 360 amino acids, in the number 6 spot, there's a substitution. The amino acid valine is put in at the number 6 spot by accident in place of glutamic acid. One little mistake. It's the only mistake in the strand. It happens right at the beginning of the strand. And yet by putting the wrong amino acid in place in the number 6 spot, the amino acid valine for glutamic acid, and they are very, very similar, but they're different. They're very, very similar in their structure, but they're very different. By putting valine in, now you end up with hemoglobin that doesn't work right. Yeah.
I was just gonna ask, isn't sickle cell beneficial when somebody has like malaria? I couldn't remember why.
Okay, good point. Yes, you can be a carrier of the sickle cell. You can have one gene, one recessive gene, or recessive trait, but not both. So you don't have disease, but you can be a carrier. And for some bizarre reason, if you have the recessive trait, you are immune to malaria. So, because malaria is one of those diseases that nobody pays attention to unless you come from sub-Saharan Africa. It kills millions of people a year. 3 or 4 million people a year die from malaria, particularly children, and nobody pays attention to it. And it is carried by a mosquito that the Anopheles mosquito, and destroys red cells. But apparently, if you have the trait, you're immune to malaria. And so you tend to survive where other people die. And yet, so you carry the trait with you, you survive malaria. So very bizarre, why does that happen? I don't know. I don't know why malaria, why an individual that's carrying the trait, now they don't usually have any symptoms of the condition. They may have a few, but they're generally can live a normal life, bearing in mind that they're still carrying that recessive trait. But if they live in sub-Saharan Africa, where malaria is prevalent all over the place, then they're immune to it. So it's very strange. You're exactly right.
I was looking at something recently, and I had mentioned it, and I was like, but I couldn't remember why that was the case. I knew it come to my head, but I couldn't remember why that, like that if you had sickle cell that or whatever carried it, that it would you would be immune to malaria.
Mm-hmm.
Which was interesting. It always is interesting to me, which is why it's more common for sickle cell to be in African American people, obviously, or whatever, because they survive to be mature, like everybody who survives gets to reproduce clearly, and then the gene is more common throughout the generations. Right.
Well, and it's actually anybody that is a descendant of sub-Saharan Africa is going to have, that's who it shows up in, not just African Americans, but anybody around the world the trait tends to show up in more people from sub-Saharan Africa than anywhere else. So, okay. So that's why it's important. That's why the order of these amino acids dictates the protein. That's why the bases are important, because the bases dictate the order of the amino acid. We have to get it right. And you've heard the term genes, right? We talk about genes. Genes are nothing more than a physical location on the DNA strand of a specific set of bases that code for an amino acid. That's all it is. That's all we have. It's that simple. The gene is nothing more than a physical location. And the good news is that we are terribly redundant. We may have hundreds of locations on our DNA strands for the same gene. We don't have, one location that says, okay, I ran out of material, no more hair color for you. You're gonna start losing all your pigmentation in your hair. It doesn't work that way. We have lots of locations. In case one place has a problem, we have other, we contain that information in other places. Because areas will wear out, areas will break down, mistakes will happen. We have an error rate in our DNA of about 1 in a billion. That's pretty good. if something happens and normally we mess up the DNA strand, we've got plenty of other places to cover it. So now when we get into chapter 3, I'll talk about when things go wrong. But basically we have a pretty, 1 in a billion is a pretty good error rate. It's pretty tiny. So the error rate there would be like 6 people out of the whole world would have the mistake. That's that's pretty good. Okay, now RNA is the other nucleic acid. The DNA never leaves the nucleus. The DNA is a double strand. It contains all of our information. Every cell in our body that has a nucleus has the exact same DNA. It's just what's turned on, what's turned off. Now the RNA takes the information. The DNA says we need to make more growth hormone. So, or we need to make more collagen. So, or we need to make more hair color or eye color or whatever. And so the instructions from the DNA will go out to from the nucleus to the interior of the cell, and the cell will use the RNA instructions to assemble that protein. And that's all it'll make. It'll go to a spot in the cell, it'll make the protein, and then it's done. And when it's done, the RNA separates down to its nucleotides and is ready to go again. And then instructions will come out from the nucleus. Let's go make hemoglobin. And so we'll make our hemoglobin. And we'll continue to do this. The DNA never leaves the nucleus. It stays there. But the RNA goes back and forth all the time, producing, controlling the actual production of the proteins we need. There's 3 types of RNA. We'll talk about that again in chapter 3. We have messenger RNA, which actually takes the information out to the cell. Transfer RNA, which brings in the amino acids. And ribosomal RNA, they form a location called the ribosome where the actual assembly of the protein occurs. Okay.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has 1 more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of the nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
I like how he says the base sequence is significant. Of course it is, because it's the sequence of the bases that determine the amino acids. You also might have noticed on the animation that we use the sugar and the phosphate as our backbone. If you imagine a ladder, the upright parts of the ladder are the sugars and the phosphates. The rungs of the ladder are the hydrogen bonds between the bases. And the spiral shape is just a function of the hydrogen bonds pulling on those bases. Okay. Now, the last type of nucleic acid is called ATP, adenosine triphosphate. This is what our cells use for energy. Our energy comes from glucose, and we will take the glucose and break it down to make ATP. We can't just release the energy from a molecule of glucose right into the cell. It would be too much, too fast. So we use that energy, we park that energy temporarily in a ATP molecule. An ATP molecule can hold energy for about 7 seconds. So we can make it, use it, let it break apart, make it again, use it again. We can recycle the ATP over and over and over again. ATP consists of 3 phosphates attached to adenine. Let me show you what it looks like. This is what the ATP looks like. It is a type of nucleic acid. It's only, it doesn't have the acid group. It has a base and a ribose sugar and a phosphate. It's a nucleotide type structure. What happens here is that in ATP, there are 3 phosphates held together. Phosphate 1, phosphate 2, and phosphate 3. Phosphate 3 is held on with what we call a high energy bond. So if we make an ATP, we park energy from glucose in the high energy bond. We force energy into that system to attach that 3rd phosphate. And that will last for about 7 seconds. And then it'll break apart. So we can't store a lot of ATP. We only have about a 7-second supply of ATP at any given time. So we're constantly making more ATP. We're always busy making ATP. But we can transfer energy around our bodies from cell to cell or structure to structure through the movement of the ATP. If we contract our muscles, our skeletal muscles are driven by ATP. Everything we do is driven by ATP. And as we use it up, we're still making it again and again and again. So it is the 3rd phosphate that is the important part of the molecule. And so there's a high energy bond between the 2nd and 3rd phosphate. When we break that bond, which we do every time we do something, we release energy. And we drop back to what we call ADP, adenosine diphosphate, and an inorganic phosphate floating around. And as soon as we have energy, we jam it back together again. It's like recharging the battery, only we have to recharge the battery every 7 seconds instead of every day. So, and that is the end, finally, of chapter 2. So, and I'm going to call it a morning. You know, we got 10 minutes to go, so we'll just call it a day here. So, now, let me, we are going to need to have a test on the first unit. And we're about right on track for that. So I'll give you the option. You can take it in class, you can take it online. Just let me know what you want to do. I am thinking that probably if it's going to be online, If you take it online, it'll open up next Wednesday. It'll be open for 2 days. If you take it in class or on the ground, you can take it. You can take it sometime after Wednesday, Wednesday or after. You can take it. I can arrange to let you take it on the ground somewhere.
This coming Wednesday?
Next Wednesday, not, yeah, I'll give you more than 2 days to get ready for it. So now I will say on Wednesday, I'm going to go right into chapter 3 on cells. So the test stops now, the test content stops with the end of chapter 2. So we'll be starting a new unit, cells and metabolism. So we'll go on. But if you want to take the test on ground, I'll arrange for you to take it with Jennifer, the tutor, or someplace, but I really don't want to use up a lot of class time for the test. But I'll give you the option, whatever is more comfortable for y'all, on ground or online. So anyway, just let me know. Let me get out of here now, and otherwise I'll use up all your extra 12 minutes I gave you off.